Hello viewers, welcome to RJ's Botany. I'm back with a new video on topic translation, that is the mechanism of protein synthesis. We have discussed in central dogma that if mRNA is formed from DNA, then this process is called as transcription. And if mRNA forms a polypeptide chain, this process is called as translation. So both transcription and translation completes the total process of central dogma. So this is here about translation, the process of conversion of genetic information in codon language. Codon, that means like AUG, triplet codon, CGA, UAA, these are all codon. It carries genetic information. So the conversion of genetic information in codon language of mRNA into amino acid sequence of polypeptide chain. That means the codon language of mRNA into amino acid sequence of polypeptide chain. mRNA contains codon AUG, CAU, like this. Next point, it occurs in cytoplasm in both prokaryotes and eukaryotes. So total translation process occurs in cytoplasm of cell. Machineries includes ribosomes, amino acid, tRNA, amino acid tRNA synthetase, mRNA, and some other factors. So these are the requirements for protein synthesis. First is ribosome. This is the cell organelle present in cytoplasm of cell. It has two subunit. One is smaller subunit and other one is larger subunit. Next is a larger subunit. It has a groove present. This one is groove. 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 And next other one is it has two side, one is P side and other one is A side. P means peptidyl site and A means amino acyl site. So ribosome has two subunit, one is smaller subunit and other one is larger subunit, and it has two site, SITE site, that is P site and A site, and other one is also E site is also present. Next is ribosome is of two types. One is 70S type and other one is 80S type. S means Sedbor unit. 40 plus 30S. Smaller subunit and other one is larger subunit. So this is all about ribosome, and another function is the function of ribosome is it helps in protein synthesis. So it is the main site of protein synthesis where protein synthesis occur, and it is also called as protein factory of cell. Protein factory of cell, as it helps in protein synthesis. So coming to the mechanism of protein synthesis, the whole mechanism of translation involves following steps like activation of amino acids, and amino acids will get activated, and it will get activated with the help of ATP, that is adenosine triphosphate, which is called as energy currency of the cell. Next is transfer of activated amino acid to tRNA. The activated amino acid get transferred to tRNA, that is transfer RNA or soluble RNA. Next step is polypeptide chain get initiated. Polypeptide chain will start. Next is elongation of polypeptide chain. It will continue. Polypeptide chain will continue, means elongates. Last one is termination of polypeptide chain. Lastly, polypeptide chain formation stops. So coming to the steps here, first one is activation of amino acids. Amino acids, there are 20 types of amino acids, and codon, like AUG triplet codon, we have discussed in genetic code. There are 64 types of codon, and for particular amino acid, it is particular codon present, like methionine, like AUG. This is a codon, triplet codon, and it codes for amino acid methionine. Methionine. Next is amino acid gets activated with ATP in presence of enzyme amino acyl tRNA synthetase. This is a enzyme amino acyl tRNA synthetase to form an amino acyl adenylate enzyme complex. See here the diagram. This is the amino acid portion. It gets activated with ATP as triphosphate is called as triphosphate. That means three phosphates are present, and A means adenosine. This is a nitrogenous base, and it gets activated with the help of enzymes like aminoacyl tRNA synthetase. This is the enzyme here, and it forms amino acyl adenylate enzyme complex. Amino acyl A is present, adenylate present, one phosphate. That means amino acyl, amino acid, adenine present, and one phosphate is present. This is amino acid adenylate enzyme complex, and the rest two phosphate here present, that is pyrophosphate. Next step is transfer of activated amino acid to tRNA. Here amino acid get activated, that means amino acid adenylate enzyme complex has been already activated, so it will be transferred to tRNA. Transfer of activated amino acid to tRNA means amino acid AMP as one phosphate is present, that means monophosphate, adenosine monophosphate, transfer to 3-OH end of specific tRNA. 3-OH end of specific tRNA means in the structure of tRNA, here one short arm is present, another one long arm is present. This one is 5-P and this one 3-OH end. So amino acid AMP transferred to 3-OH end of specific tRNA and linked through ester bond. This is the structure of tRNA, and the activated amino acid adenylate enzyme complex will get attached to 3-OH end of tRNA with the help of ester bond. This is the ester bond. Ester is what? Bond COH bonds to OH group with the help of enzyme. That the enzyme present is amino acid tRNA synthetase, as tRNA is attached here. So the enzyme is amino acid tRNA synthetase. See here the diagram. This is the structure of tRNA. This is the ester bond, and here the amino acid get attached. So amino acid get attached to tRNA with the help of, that is, ester bond. Okay, so this is the step here, the transfer of activated amino acid to tRNA. Activated amino acid get attached to tRNA with the help of ester bond. Next is, here specific amino acid recognized by DHU arm of tRNA. That means DHU arm means this portion is the dehydroxyuridine arm. DHU means dehydroxyuridine arm. Its function is it will recognize specific amino acid. So in tRNA structure, DHU arm will recognize specific amino acid which will get attached to tRNA, 3' end of tRNA. So here amino acid AMP enzyme complex tRNA, that is 3' this is the structure of tRNA, and the enzyme present is amino acid tRNA synthetase. Here amino acid get attached to tRNA by ester bond, and the rest part is adenosine monophosphate, that is AMP. Next, third step is polypeptide chain will initiate. That means polypeptide chain will start. Protein synthesis starts with amino acid methionine in eukaryotes and formylmethionine in prokaryotes. So I have said you that AUG codes for amino acid methionine which is present in eukaryotes, and formylmethionine in eukaryotes, which is coded by initiation codon, that is AUG. So AUG codes for amino acid methionine. It is called as start codon or initiation codon. Start codon or initiation codon. So in polypeptide chain, AUG is the starting codon and it is fixed. Next is there are three initiation factors in prokaryotes, like IF3 means initiation factor 3, 2 and 1. Another point, remember here, ribosome are present in the cytoplasm of cell. Ribosome are present in the cytoplasm of cell, but the subunits present in the ribosome are separated in this are present in separated form in cytoplasm. That means both larger subunit and smaller subunit are separated, and it will remain together during protein synthesis. So the smaller subunit here, the structure is the mRNA present here. This is the structure of mRNA. Next, if the first codon is A, U and G, the next one will be if C, C, U. Here here thymine will be not present, because as we have read in RNA, the nitrogenous bases are adenine, guanine, cytosine and uracil. So the start codon is AUG, and the smallest subunit of ribosome, that is 30S, attaches to the mRNA covering the initiation codon with the help of initiation factor IF3 and magnesium ion. That means in mRNA, first smaller subunit will cover the mRNA. That means here this is the smaller subunit. Next point is tRNA containing formylmethionine attaches with initiation codon of mRNA with the help of initiation factor, that is IF2. It is said in case of prokaryotes, and energy source is GTP. And the next step is larger subunit of ribosome covers the smaller subunit, and F-met tRNA means formylmethionine tRNA reaches P site, that is peptidogene site of ribosome, and A site remains empty and require factor IF1. See here, first mRNA in mRNA, smaller subunit covers the mRNA. Next, the larger subunit will cover the mRNA. Here, one groove is present. I have said you one groove is present. This one is groove, and this one is B site, and this one is A site. Here, tRNA is present in the outside, not in mRNA. It is present in the outside, and it has one T amino acid. First tRNA is present with first amino acid. If the tRNA will reaches the ribosome site, protein synthesis site, it will covers the P site. First tRNA will come with amino acid to peptidyl site, and A site will remain vacant. Okay, next point is, in next step, the largest subunit of ribosome covers the smaller subunit, I have said before, and forming methionine tRNA reaches P site, that is peptide site of ribosome, and A site remains empty and requires factor IF1. See here the diagram. Here the 30S, that is the smaller subunit, 50S, this is the larger subunit. Here tRNA is present with amino acid, first tRNA with first amino acid. See here, this is the groove present, and AUG, this is one codon present. In another portion there is another codon present. This is the P side and this is the A site present here. And the first tRNA will covers the P site of ribosome. First RNA will attaches with first amino acid and will covers the P site of ribosome. Here, the initiation factor IF3 helps when smaller subunit covers the mRNA, and IF2 here helps in tRNA to covers the P site, and IF1, that is, it helps in IF1, that helps to cover the larger subunit over the smaller subunit. Next diagram is, next heading is elongation of polypeptide chain. Here, tRNA containing second amino acid attaches to second codon of mRNA. Here, tRNA present with second amino acid with the help of elongation factor, that is EF factor, and GTP guanosine triphosphate. So, same here, the first tRNA present with first amino acid and second tRNA present with second amino acid. That means I have said in the first last step that first amino acid with first amino acid will covers the P site and A site will remain vacant. When the in the elongation portion, the second tRNA with second amino acid will covers the A site. That means the P site and A site is covered with tRNA and amino acid. First tRNA with first amino acid and second tRNA with second amino acid. Next step is the enzyme peptidyl transferase present in P site of ribosome forms a peptide bond. Here, peptidyl transferase enzyme present in P site, this one is A site of ribosome, forms a peptide bond. Peptide bond is formed between two amino acid. So here first amino acid present, second amino acid present, and this enzyme present in P site forms a peptide bond, will form a peptide bond in between two amino acid. That means in between carboxyl group of first amino acid and amino group of second amino acid. So first amino acid get free by hydrolysis of ester bond by same enzyme. Here, first amino acid get free by hydrolysis of ester bond. I have said before, tRNA is present with amino acid, but it is attached here. It is attached with the ester bond. So the function of tRNA is only it will transport the amino acid to the site of ribosome. Here the first amino acid get hydrolysis. That means the ester bond will break. It will get free by hydrolysis of ester bond by same enzyme. In translocation, here the amino acid gets separated from what? tRNA. Here the amino acids are bonded by peptide bond and tRNA get separated. Next point is in translocation. Translocation means what? Movement. Ribosome complex moves one codon ahead and over mRNA with the help of enzyme translocase. After hydrolysis of ester bond, ribosome will move one codon ahead. One codon means three nitrogenous bases ahead. Here's the ribosome covers first codon, next second codon. After bond is formed, ribosome will move one codon ahead. That means if ribosome moves one codon ahead, it will cover this portion. Here, first codon is left behind. Here the first P site and this one is A site. Next, in translocation movement process, ribosome complex moves one codon ahead over mRNA with the help of enzyme translocase. First tRNA remains outside of ribosome. If ribosome will move one codon ahead, then the first tRNA will remain outside of ribosome with amino acid without amino acid, and second tRNA occupy P site and third amino acid tRNA get ready to occupy A site. That means if ribosome will move one codon ahead. That means in first position this was the P site and this was the A site. But after moving one codon ahead, this will be the P site of ribosome and this will be the A site of ribosome. So P site occupy second amino acid occupy P site and third amino acid get ready to occupy A site. That means first amino acid is outside of ribosome. Here the second amino acid which was present before is present in P site, and this portion is what? Vacant. This position is vacant. That means amino acid site is vacant, and third tRNA with third amino acid is will get ready to occupy the A site of ribosome. Here see the diagram here. tRNA is present without amino acid. Ribosome has moved one codon ahead. Here this one is P site and this one is A site. Second amino acid occupied the peptidyl site which was present before amino acid site, and the A site, that is amino acid site, this one is vacant. Now the third amino acid tRNA will get ready to occupy the A site, this portion. Likewise, ribosome will move one codon ahead throughout the mRNA. Termination of polypeptide chain. After reaching the termination codon of mRNA, here the protein synthesis will stop. Likewise, amino acid will form a peptide bond with amino acid. So this chain is called as what? Polypeptide chain. In termination portion, polypeptide synthesis stops at ribosome reaches the termination codon. In mRNA, first codon is AUG and the last codon, the termination codon or stop codon, or it is also called as nonsense codon. It may be UAA or UAG or UGA. These three are stop codon, termination codon, or we can call it as nonsense codon. So, the enzyme peptidyl transferase along with release factor, as it will get released, that means the polypeptide chain which is formed will get released. Hydrolysis ester bond of last amino acid. By same process, the ester bond will get free from amino acid and tRNA. Finally, the polypeptide chain is released from ribosome mRNA complex. Finally, ribosome reaches the termination codon. Likewise, amino acid is bonded with peptide bond and the last tRNA present with last amino acid will are present in P site of ribosome. After that, ribosome will be outside and tRNA will go out and it will form a long chain of amino acid, that is polypeptide chain. When two or more polypeptide chain are formed, then it forms protein. So, amino acids are called as building blocks of protein. Here, as polypeptide chain is formed, so protein synthesis happens. So this is all about the process of protein synthesis. Initiation occurs, elongation next, termination occur. So here in we have read in definition that the genetic information codon language of mRNA into amino acid sequence of polypeptide chain. Here the amino acid sequence. This is the amino acid sequence of polypeptide chain. This is the polypeptide chain is formed and it occurs in cytoplasm of cell. So I hope you like this video. Thank you.